Bienvenido. I hope I said that correctly, Julia. You can correct me if I'm wrong. Bienvenidos, bienvenue, and welcome. So it's my great pleasure to host the basic science session today, hosting two speakers, one from Brazil and one from Mexico. And so, of course, we all have a newfound appreciation for RNA biology in 2022, given all of the amazing breakthroughs for the COVID vaccine that happened. And I myself am dipping my toe in translational control through collaborations with leaders in the field, such as Dr. Nachum Sonnenberg and Dr. Ivan Topasevich, who gives his regrets he can't be here today. He's flying around in Japan today, giving a talk. So I'm hosting on his behalf. And the first speaker will be Dr. Is it Glaucia Hajj? The first, yes. Dr. Glaucia Hajj, she is an expert in translational control. She received her PhD in biochemistry in 2004 from the University of Sao Paulo. In 2010, she joined the AC Camargo Cancer Center in Brazil, and she studies the intersection between cell signaling and control of mRNA translation in fatal tumors such as glioblastoma, which we will hear about today. She has published over 40 articles in highly qualified journals and received the Highly Qualified Personnel Award from PrioNet Canada in 2010, and a bunch of other notable prizes with her efforts in trying to find new druggable targets in cancer. She is a leader in her field and as such has strong collaborations with researchers at McGill University, the Karolinska Institute in Sweden, and the University of Eastern Ontario. And so without further delay, we will hear from Dr. Hajj on translational control in human glioblastoma. Thank you so much.
Hi everyone. Thank you for, at first I would like to thank the organizers for the invitation to talk to you today. My talk will shift a little bit in theme, and you're gonna hear a little bit more about basic science. I have tried to not to include a lot of boring details, but you can ask me for whatever doubts you may have. Sorry, just checking if this is okay. Okay, it's okay. So I work at the AC Camargo Cancer Center, which is located in São Paulo, Brazil. And it is a cancer-dedicated hospital that also promotes high-level education and research. Our research institute is dedicated to basic, clinical, and translational research in cancer. In my laboratory, we have been studying mechanisms of translational control in cancer for the past few years. And today, I will focus my presentation on a high-throughput technique that adds a new layer of information into the understanding of complex biological problems, such as cancer. So for the past 2 decades, we have faced a revolution in the ways that we study and understand cancer. The advances in nucleic acid sequencing and bioinformatic techniques have provided the scientific and medical community with an enormous array of information in the molecular characterization of cancer. Multi-institutional initiatives, such as the TCGA, have now gathered and organized molecular information of thousands of different tumors and present us with a complex multi-layer information. In most TCGA projects, we are able to view information on DNA structure, such as mutations and copy numbers. Another essential aspect in the molecular characterization of tumors is the identification of gene expression profiles. And this is generally obtained by sequencing steady-state RNA levels. However, it is generally underestimated that RNA translation has a fundamental role in gene expression. Because the RNA needs to be processed, transported, and may be stored or translated, total RNA levels obtained in the transcriptome do not necessarily match the expected proteome. If we identify, for example, the complete proteome or the transcriptome of a given cell, we can observe that the average number of proteins per gene is around 50,000, which clearly cannot be explained only by the average of 19 RNA molecules per gene. As a consequence, if we plot the correlation among protein and RNA, only 40% of the proteome can be correlated with the correspondent RNA levels. For example, while a gene can be abundantly transcribed, its translation rate can be reduced. So the total protein synthesis would be reduced as well. The opposite is also true. A gene whose translation, the opposite is also true. A gene whose transcription is reduced or remains the same can be upregulated by translational mechanisms. So translation adds another layer on the regulation of gene expression. So that being said, can we determine gene expression profiles by using differentially translated genes instead of total RNA? And if so, would this give us additional information on the biological behavior of tumors? To answer that question, we use the technique called Polysome Profiling, which allows us to biochemically separate the messenger RNAs that are being actively translated. This technique takes the advantage of the fact that actively translated RNAs are coupled to one or more ribosomal subunits. Since these are massive structures composed of dozens of proteins and RNAs, their large size allows for the separation by sedimentation in a sucrose gradient. After the biochemical isolation of the translating RNAs, they can be identified by RNA-seq. We started our project in glioblastomas. This tumor was chosen because despite of being the target of extensive molecular characterization through the last decade, no advances in targeted therapies have proven efficient, and it still represents one of the deadliest tumor types. Glioblastomas are one of the most frequent tumors of the central nervous systems in adults, and they have an astrocytic origin. As I told you, they present a very poor survival and very limited treatment options. In our first pilot study, we received a tumor fragment of large size that was histologically very heterogeneous. And we were able to separate this tumor in 8 different fragments that could be histologically classified from a low grade to a higher grade. After performing the biochemical separation of the polysomes, the first observation that we can take is that the levels of translation are increased in high-grade fragments when compared to low-grade, which makes sense if we think that they're more metabolically active. Then we identified the genes present in both total and translated RNA. And this gives us 3 possibilities of gene expression regulation: abundance, buffering, and translation. When a gene is regulated both by transcription and translation, this was called abundance. When there is no impact of transcription, the regulation mode was called translation. And when an increase in transcription is not followed by translation, this was called buffering. So when we plot the expression values of both total and translated RNA from high-grade versus low-grade fragments, we can identify these different modes of regulation. And we can observe that nearly half of the genes identified have a translational component of expression regulation, which means that half of the information would be missed if we had measured only the total RNA. When we compare biological processes associated with both total and translated RNA, we can see that in translated RNA, 3 biological processes associated to cancer cannot be found using total RNA. Response to stress, epithelial to mesenchymal transition, and immune system processes. With the success of our pilot study in mind, we proceeded to perform translatomic in a larger scale. And to do that, we initiated with a cohort of 17 glioblastoma cases, whose total and translated RNA was biochemically isolated and identified. Then, a second set of 20 samples was used afterwards for validation of the results. In here, I show you the first set of results, where we successfully identified both total and polysomal RNA from the first set of 17 samples. The data is presented here in a PCA plot, which groups the samples according to their similarities. So we can see here that the polysomal samples are grouped together and the total samples are grouped together. This means that this set of samples is more similar among itself than with the correspondent total samples. When polysome RNA was used to perform unsupervised clustering of the samples, we observed a separation in 3 groups, with a striking difference in survival probability, as evidenced here in this Kaplan-Meier plot. However, if we use total RNA to perform the same analysis, we observed a very different result. The separation in groups is not so evident here, and the impact in survival is not observed. In here, we can also observe that the samples that were grouped together using the polysome RNA are scattered when we use the total RNA information. Also, if we compare the clusters generated by polysome RNA with molecular clusters indicated by the TCGA, we can observe that there is no match. However, the molecular clusters defined by the TCGA do not present differences in survival, whereas our data support an impact in the clinical setting. If we plot the total versus translated RNA from the 3 groups to evaluate the modes of gene expression regulation, we can notice that there is a significant number of genes whose expression is modulated only by translation and not by transcription. We then classify the differentially translated genes in gene ontology groups that represent biological processes. The first cluster was called MIT, as in this group, most upregulated genes include the ones that code for mitochondrial proteins. Many cancer cells upregulate mitochondrial processes in order to provide the substrates required for synthesis of macromolecules, such as lipids and nucleotides. Multiple substrates feed into these biosynthetic pathways, thus providing cancer cells with metabolic flexibility to support growth. An altered vasculature is a characteristic of glioblastomas. However, the mitochondrial electron transport chain is able to function optimally even at oxygen levels as low as 0.5%. Thus, a pharmacological inhibition of the electron transport chain with drugs such as metformin, for example, could be an interesting target for this group. Metformin has been tried in multiple clinical trials as an anti-cancer agent in combination with standard of care therapies. The second cluster was called TOR, as this group most upregulated genes include those translationally regulated by mTOR and also angiogenesis-related genes. The relationship between mTOR, angiogenesis, and cancer is extensive, and several inhibitors of these processes are approved for cancer therapies. Indeed, mTOR inhibitors were tested in glioblastoma clinical trials with poor results. Our data, however, suggests that only a subgroup of patients would benefit from this therapeutic strategy. The third cluster was called SIL, as most upregulated genes in this group include those related to cytoskeleton and primary cilia regulation. Almost all cells have a specialized cell surface projection called the primary cilium. Although these structures were first described more than a century ago, the full scope of their functions remain poorly understood. In addition to their well-established roles in mechanosensation, primary cilia are key participants in intercellular signaling, and their alterations are involved in many human disorders, such as cancer. In glioblastoma, they are associated with resistance to temozolomide treatment. To validate our findings, we created a molecular signature that was applied to the translatome of a second cohort of patients. This molecular signature was able to classify the additional set of samples into the previously established 3 groups that also presented a large difference in survival. In conclusion, we described for the first time the translatome of human glioblastoma that yielded the separation of the samples in 3 molecular groups with impact in survival and activation of different molecular pathways, suggesting that a difference in treatment options could be beneficial for patients of the 3 molecular groups. I want to thank all the people that participated in this work from the Camargo and from other collaborating institutions, our funders, and also our most recent collaborations in Canada that will allow for the continuation of this work in the validation of some of the targets that we found here and the expansion of this work to other tumor types. Thank you for your attention.
Beautiful talk, Glaucia. I really enjoyed that. Thank you so much. So any questions from the group? If not, I can start with questions. So Glaucia, you showed this beautiful data that in the higher grade glioblastomas, you have more active translation as measured by your Polysome profiling, which is amazing. So my first question is, which type of inhibitor? So there are inhibitors of the 4F complex against 4E, 4A that have been developed. Do any of these cross the blood-brain barrier, and how do you see these being used clinically?
So the advantage of glioblastoma is that the vasculature is very poorly organized. So in these tumors, the patient has a breach in the blood-brain barrier. So most drugs could actually be used to treat these patients due to these problems in the vasculature. I'm sorry, I didn't hear you.
Sorry. So since there are so many drugs that are able to inhibit translational control, in your samples, have you been able to probe? Is there like an overexpression of the helicase, the scaffold protein? Have you looked at that at all? Sorry, we can't hear you. Yeah, okay.
Okay. We have been looking. We didn't see a specific increase in many modes of translation regulation. We saw a big difference in mTOR related mechanisms and a few proteins of the translation machinery that I don't recall in memory, but there was not a general increase in the machinery.
Muted. They're going back and forth, muting you and me at a time. Okay, Glaucia, so I guess if there are no more questions, oh, there's a question here. Go ahead. Thank you.
Thank you, Dr. Hajj, for your wonderful talk. My name is Luisa Bonilla. I'm a medical oncologist in Northern Ontario. And as you probably know, right now, the way that we choose medications for our cancer patients are based on mutation status, which is a very, very primarily way to decide about treatment. It seems to me with this new technology that you are introducing, it will be more effective to actually choose medications based on the translatome and choose those patients that really will benefit from this. Which are the next steps in you extending this to other treatment or to other types of cancers and extending the use of this? Thank you.
Thank you for your question. So our next steps are on breast cancer. We have a project that already started that we're doing 200 samples. And also on clear cell renal cancer, we are also, we have like approximately 80 samples that we started the project. Unfortunately, it takes time because it is a labor-intensive methodology and also a bit expensive because everything that you have to sequence twice for the total and for the polysome RNA. In regards to glioblastoma, we really want to compare our results with the main DNA alterations that we observe in these tumors, like if there's a deletion or if there's any mutations. We want to compare our results to the mutations and see if there's, or which is the relevance of the alterations. It is a bit harder because that may require a larger cohort, and it is not always easy to find because we also use a fresh or frozen tissue, and that reduces the ability of finding the samples to perform the studies. And we also want to validate our results in the sense that we expect a much higher correspondence to the protein levels. So we want to choose a set of targets and validate this using the protein in immunohistochemistry.
Perfect. Thank you so much, Dr. Hajj, for your wonderful talk. I have so many more questions, but probably I should email you. So thank you so much. And we now invite the next speaker. So it's my great pleasure to introduce Dr. Greco Hernández Ramírez. He is from Mexico, and he has received his PhD degree from Madrid, Spain in 1992. He then did 2 postdoctoral fellowships, one at the Max Planck Institute in Germany and the other at the University of Bern in Switzerland. And then he moved to Canada to pursue more research in RNA biology under the mentorship of Drs. Paul Lasko and Dr. Nachum Sonnenberg here at McGill University. In 2011, he was appointed principal investigator of the National Institute of Cancer in Mexico. He has published many articles in top journals, including one in the latest issues of PNAS, and his lab is broadly focused on 3 lines of investigation. One, in developing therapies against prostate cancer that target using therapeutic mRNA vaccines. The other is to uncover novel mechanisms that can confer resistance to cancer. And finally, studying mRNA features that encode different oncoproteins involved in cervical cancer and glioblastoma. So thank you, Dr. Hernández Ramírez, for speaking here today. Thank you.
So hi everybody. I don't see. So how should I change slides? I'm not sure.
Hi, Dr. Hernandez. You should just be able to move the slides along, or can you not see the next slide on the?
Okay. So hi everybody. I'm glad to be in this meeting today. I want especially to thank Professor Ivan Topisirovic for the invitation to share with you some results from my lab at the National Institute of Cancer. My name is Greco Hernández. I also thank Sonia del Rincon, Ben Howard, and the organizers of the meeting. Just before all my talk, I want to say that I am happy. I am still in touch with Canada and the different researchers I met in Montreal during my years in the Nachum Sonnenberg and Paul Lasko labs, including Ivan. So today we'll be talking about 2 projects related to problems of paramount interest for the public health in Mexico. Cervix cancer and prostate cancers. As the previous speakers, I won't be so in detail with the techniques about the biochemistry we were doing. I tried to avoid the jargon and to go deep inside biochemistry reactions. So, because I know the nature of this audience. Let me see if I can change my, I don't understand how should I change.
Do you want to you could just share your screen if you can.
How? Share screen?
Present. Yeah, if you go to present at the bottom, there should be an option if you click on that to share screen.
Okay. Seleccionar pantalla. This is the screen. Okay.
If not, I can share my screen and you can just let me know when to change slides.
It's not working. I knew this is going to happen. So no, I understand. So is diapositivas. So it's not working, right?
No, but I can share my screen with your slides, and then you can just let me know when you need me to switch slides.
Okay. Yeah.
Okay. Perfect.
Please do it because I'm not understanding how this. So now everything is okay.
Yep. Yeah. So you can go ahead and just say next slide whenever you want me to change the slide.
Okay, so I was saying that I will be talking about 2 projects related to problems of paramount interest. So for the public health in Mexico, cervix cancer and prostate cancer. According to the World Health Organization, prostate cancer is the most prominent cause of death among males. In women, breast, thyroid, and cervix are the 3 most important causes of death by cancer. Here I am showing the numbers of new cases for 2018 that I believe is the most updated number of cases and death in cancer by the World Health Organization. Next, please, next slide. So one of the interests of my lab is to study the gene expression at the level of mRNA translation of the human papillomavirus, which cause cervix cancer. There are about 220 HPVs, or human papillomavirus, described, but only few are considered as high-risk viruses. In particular, about 70% of the cervix cancer cases are due to persistent infections by the types 16 and 18. Both types belong to the genus Alpha. So I have to say that in the right part of the slide, I am showing the genome of the HPVs. High-risk HPVs encode two potent oncoproteins, termed E6 and E7. Next, please. Next slide. So the interaction and activity of E6 and E7, the two potent oncoproteins, lead to the inhibition of apoptosis and DNA repair, and to promote cell cycle progression and proliferation. Next slide, please. Next. Early studies in culture cells during the decade of 80s show that the E6 mRNA of the high-risk HPVs possess very short UTRs. So I am showing here the open reading frame, and the 5' UTRs. So this part of the molecule, of the mRNA molecule, is called also leader. So these observations were done using different techniques, as I am showing here: RNase protection, primer extension, 5' race, for example. And this is and I am here showing the case of HPV 16 mRNA, which encode the E6 protein. Next slide, please. So in the case of HPV 18, these examples are the HPV 18. This feature is even more extreme. There are 5' UTRs populations with 9, 7, 3, and even 0 nucleotides long. As I said, all these experiments were done in culture cells. Next slide, please. So high-risk HPVs, in particular, the type 18, expressed leaderless mRNAs. This was very intriguing because a typical mRNA contains 5' UTRs longer than 200 nucleotides across eukaryotes. In many oncogenes, the leader is even longer. For example, many oncogenes, they have 300 nucleotides long 5' UTRs or leader. And the case of picornavirus is even more extreme because they contain UTRs kind of 600, 800, up to 1,200 nucleotides long. So this situation of the potent oncoproteins was very, very intriguing and very interesting to start to study. Next, please. So as we know, protein synthesis happens when translation machinery is recruited to the 5' end of the mRNA, which scans along the UTR to reach the start codon. Next, please. Next. Yeah, the whole process can be inhibited by the action of 40BP, the protein called 40BP or 40 binding protein. So next, please. The questions we asked were, what is the size of the E6 mRNA 5' UTRs in tumors? Because as I said before, during the decade of 80s, all experiments were done using culture cells. So we wanted to know whether this is the case in happening in tumors, in actual tumors. And the second question we asked was, how does this mRNA is translated? I mean, what factors are being used by these weird mRNAs to be translated during virus infections. Next, please. So we isolated HPV mRNAs from many tumors, observing that the high-risk HPVs indeed possess extremely short or even lack a 5' UTR. The most representative were these two UTRs isolated from up to 11 different cervix tumors. So I am talking about the HPV 18. So in the lower part of the 18, we see a lot of mRNAs without a 5' UTR. So in this case, we analyzed 11 tumors, but we kept doing those isolation and those experiments, and we saw the same situation. Next, please. To study how E6 mRNA is translated, we undertook an in vitro translation approach using synthetic mRNAs, encoding the firefly luciferase as a reporter cistron and cell-free lysates. These mRNAs were synthetic mRNAs prepared in the lab, so we could control the size and the length of every part of the mRNA, including the 5' UTR that was the subject of our study. Next, please. We constructed several mRNAs with UTRs of three nucleotides or nothing at all, and compared their translation and, as I said, in vitro translation assays. As we can see, the 0 and the 3 nucleotides UTRs were very well translated, but not the luciferase control, lacking an UTR. These experiments were done not only in HeLa but in different human cells. As you can see in the black parts, the 3 nucleotides long and the 0 nucleotides long UTRs promote translation comparable to the control containing a 15 nucleotide long UTR. Next, please. Next slide, please. Can you show the next, please? No, it's not working. Next slide, please. Hi. So we test translation in the absence either of or a polyethylene. Molecules I'm showing here are needed for the translation of a regular mRNA across eukaryotic species. So we prepare mRNAs. No, before this one before. It's not working.
Hi, Dr. Hernandez.
So can we show the previous slide, please?
Yeah, would you be able to turn your video off? I think you're cutting out maybe due to internet.
So we can experiment to evaluate the dependency on 4E by adding recombinant 4EBP to the translation reaction, observing also inhibition of translation. So as I said before, the protein that binds to 4EBP, 4E binding protein, is able to block the whole protein synthesis process by 4E. So as we can see, in the graph, every time that 4E recombinant have translation is abrogated. And we also evaluated the dependency on 4E, and as we can see in the right plot, in vivo cells were knocked down to deplete for 4E protein, endogenous protein, and we see also that the translation of those mRNAs were abrogated. was abrogated. So we also evaluated the dependency on the RNA helicase for A. To do that, we added recombinant for A to the reaction, an increase in the presentation of all mRNAs. We also performed reactions in the presence of 4A inhibitor. We observed a translation inhibition of all mRNAs, but not controlled. So in the left graph, I am showing translation increase
Greco, ¿me oye?
because we added recombinant 4A protein, the helicase, that is involved in the scanning process of translation. Plot I'm showing the addition of a specific inhibitor of the helicase. As a control, we have the HCV-IS mRNA that is not dependent on 4A or 4E to be translated. And next, please. Next slide. Can we show next, please? Okay. So we finally compare the sequence of the different HPVs, and we observe that the high-risk types lacking UTR share a conserved sequence within the open reading frame that we share a translation initiation of leaderless mRNA. So as we can see in the upper part of the slide, upper left, we have in red all viruses belonging to the alpha 7 species. All these viruses are high-risk types. And as we can see in the gray box, we all share this motif that we call the translation initiation of leaderless mRNA. And specifically, we see that the mRNA of these viruses lack a 5' UTR or contain a very short 5' UTR. So we mutated, as we can see in the right part, we mutated either the codon, the second codon, the third codon, I am showing in red, the 4 codon of this motif, the second and the third codon, or the nucleotide number 4, or the upstream codon of the motif we discovered. Every time we mutated any of these codons, as we can see in the lower graph, we see an abrogation in the translation of the luciferase reporter mRNA. So next, please. So we have some conclusions. Of course, I didn't show all experiments we did to corroborate every experiment, but okay, we can say that HPV, high-risk HPV 18, 14, and 39, E6 mRNA lack a 5' UTR in tumor. So this was seen by the first time because all the time experiments were done in culture cells. We can also conclude that HPV 18 E6 translation needs a novel motif to be translated, and I am showing the sequence of this motif. We can also conclude that HPV 18 E6 mRNA requires a poly-A tail, a cap, a molecule, EIF4E and EIF4A factors for translation. Well, as Sonia mentioned it at the beginning of the talk, this work was published recently, last year, in PNAS. So we were very happy for that. And next, please. The next slide, please. Okay, so now I am going to be talking a little bit about the other subject I was talking about in the beginning of my talk, that is the prostate cancer. Prostate cancer is very, very important in not only in Mexico, but the world, and specifically in Mexico, prostate cancer is the first cancer incidence in males, is the first cause of cancer males in Mexico, and not only in Mexico, but in other countries, specifically in South America. Next, please. Next slide. Can we show the next? Okay. Hello. Next. Next. Hello.
Hi, Dr. Hernandez. Which slide would you like? The
hello. Yes, please. Next slide, please. Okay. So I am showing in green, the incidence of prostate cancer is pretty important. We can see the whole American continent and most, at least, this many countries in Africa. Prostate cancer in males is the first cancer by incidence.
Hi, I think Dr. Hernandez just dropped out completely for internet.
So we're going to move on with maybe Dr. Del Rincon taking questions.
Yes, so if there are any questions, hopefully Dr. Hernandez Ramirez can join us again. But obviously this is an important area of research that requires more hands on the ground. So we are doing our part in melanoma as well. I think, we saw 2 examples of cervical cancer and glioblastoma being potential targets for novel drugs of translational control. If there are any questions, I might be able to answer them on Dr. Greco Ramirez Hernandez's behalf. Any questions from the group? No, I do have questions for him, but maybe we can then close the session and go on to the next speaker if he's not going to hop on. I'll wait for the organizers to prompt me on that end. Yeah, it's wonderful that we can use these virtual platforms, but I always say they can be a blessing or a curse. I just feel bad that we dropped him completely, or that his internet gave out. All right.
Yeah, we try to do our best with technology, but sometimes, especially in such a global meeting, it doesn't fulfill our expectations.
Okay, so then I guess I'll just close and thank everyone for their participation in the basic science session, and we look forward to future collaborations between basic scientists and clinician scientists around the globe. Thank you.